You can ask questions, but we're being recorded, so don't say anything inappropriate. So, I'm looking at you. So, what do we remember about the structure of DNA? Or I was go for smaller. What's the DNA made of?
Deoxynucleic acid.
Okay, so deoxyribose nucleic acid. What do we call the units? Like the individual units are called what?
Base pairs?
Close. Thank you. Nucleotide.
Base pairs.
So the unit is a nucleotide. A nucleotide is made of three. There's three parts to a nucleotide.
Sir, can you move
Okay,
up, sir? Thank you.
I'm gonna zoom in, zoom out, and I'm gonna it's recording. Okay. So all you gotta do is listen and take notes. Okay. So nucleotides are made of. There's three parts. There's three parts to a nucleotide, right? I heard one. I heard ribose. So we have ribose, which is this is a 5-carbon sugar. So I just write 5C in the middle, and then I'm hearing it. What'd you say? Okay, so this is the nitrogen base. And the third part to the nucleotide is phosphate group. Okay. A phosphate group it's usually P and it's got 3 O's on it, and usually they're they're usually negatively charged. Sorry, that's backwards, but O negative. So there's like three oxygen atoms on a phosphate on a phosphorus atom. That's your phosphate group. So this is our nucleotide. The difference between DNA and RNA is the deoxy. So we have deoxyribose and ribose nucleic acid. So on the ribose at the bottom, there is 2 OH on the bottom. So the deoxyribose is missing the one on the left. So this would be ribose nucleic acid. So I just drew a RNA nucleotide. If I erase this, if I erase the OH and make it an H, now I've just drawn deoxyribose. They're probably not going to ask you that minor detail, but you should know deoxyribose versus ribose. Okay? So there's 3 parts to the nucleotide. There's 3 parts. The phosphate group, the 5-carbon sugar, and the nitrogen base. Now let's compare DNA, RNA. So we have DNA and we have RNA. So I'm going to make a T-chart. D for DNA, R for RNA. So the first thing is the sugar. So in DNA, it's deoxyribose is the sugar, and RNA is the ribose. Other known differences.
One is double strand, one is single strand.
DNA is double-stranded, double strands. RNA is single strands. What about the bases?
DNA is ATGC,
Okay.
the other one is AT.
So we have adenine, thymine, cytosine, guanine. I'll write them over here. Okay, here's the bases. Adenine, so we can add ADENIN. Cytosine, CYTOSINE. Guanine, GUANINE. And T is thymine. And the last one is uracil. Uracil is RNA. Okay? And thymine is DNA. Adenine, cytosine, guanine. These three are DNA and RNA. Okay? If we're referring to them in generic terms, this is good to know. We just simply say DNTPs. This is like, and I'll explain when I'm talking about DNA replication. This is a generic word for, it's just, N is for ATCG. So when we're talking about DNA replicating, and there's different, we don't, instead of saying ATCG, just say you have a polymerase will take a free DNTP and add it to the DNA molecule. So it's just a generic term for referring to A, T, C, and G together. So the EN just stands for ATCG. Okay? So it's just good to know that acronym. Okay? When we talk about gene sequencing, we refer to DT and TPS. And we'll refer to these when we talk about gene, how we actually sequence DNA. So just know those are generic terms for ATCG. All right. Was that confusing, the DNTP thing?
Yes.
Yeah?
No.
Okay. No, yes, okay. DNTPs just refers to all the bases. So they're free-floating nucleotides in water in the cell. Okay. Okay, so back to DNA, RNA. So in DNA, we have ATCG. RNA is AUCG. Any other differences?
U and T. Any other difference? Another difference?
One is
They're both-
Euro kind or something.
DNA is built different.
There are three types of RNA.
mRNA, tRNA,
There are three types of RNA. As far as location goes, this is where you play the politician and say generally. Generally, DNA is in the nucleus. Right? If I'm talking about a prokaryote, prokaryotes don't have nucleus, they have a nucleoid. So prokaryotes don't have a nucleus, right? So generally there's a region where DNA is stored. The nucleoid in a prokaryote or a nucleus in a eukaryote. Okay? RNA tends to be where? If it's not in the nucleus, RNA tends to be in the
The low weird, the low wonky things.
what's the area that's not in the nucleus that's in the cell? Well, you're thinking organelles, but it's not an organelle.
cytoplasm.
Cytoplasm. So generally the RNA.
Sorry, Kang. Just concentrate and memorize.
Okay. And this will help us when we talk about evolution. To know this now, this will help you in evolution. RNA can self-replicate, DNA cannot. So RNA can self-replicate. Okay? DNA cannot self-replicate. There must be protein. So in evolution sense, this is the chicken and the egg problem. What came first, DNA or protein? We don't know. Because you need protein to copy DNA. You need protein to read DNA. DNA by itself does nothing, but you must have DNA to do everything else as well. Right? So what came first, we'll never know. Okay. The three types of RNA, let's go to that.
mRNA, tRNA, RNA.
So there's tRNA.
mRNA,
I don't know why I'm doing that. Okay, tRNA, mRNA, and rRNA.
What's the difference between What's the difference between mRNA and mmRNA?
There's double N.
Shut up.
I've never heard of mRNA.
What's
Major
MMRNA?
messenger. Major messenger RNA.
Never heard of it.
So there's minor messenger?
Did I just discover something? Am I going to graduate IB? I'm gonna write a
No,
thesis.
you can't graduate IU because you discovered something.
Don't need to graduate.
So, do you remember ribosomes?
Yeah.
What do ribosomes do?
Produce protein.
Ribosomes make protein, yes?
Oh my god.
And there's 70S ribosomes, 80S ribosomes.
Yeah.
Sound familiar? Okay, so there's two sizes of ribosomes. There's 70S ribosomes, there's 80S ribosomes. 70S ribosomes are where? Prokaryotes or mitochondria and chloroplast. You're being recorded, don't forget.
That was
70S ribosomes exist in mitochondria, chloroplast, and prokaryotes. Eukaryotes have 80S ribosomes. So, a ribosome looks like this. Oh, I'm going to change color. A ribosome looks like this. You have a large subunit and you have a small subunit. The small subunit, so this is, the small subunit is either 20S or 30S, and the large subunit is 50S. So if you are a prokaryote, you have the 50 and the 20. If you're mitochondria or chloroplast, so 70S ribosomes, these are going to be in your prokaryotes, mitochondria, and chloroplasts. And eukaryote, 80S ribosomes are for eukaryotes. Sandwiched between the two halves of the ribosome is going to be your messenger RNA. Okay? The ribosome is made of RNA and protein. So it's not just RNA that makes up the ribosome. So ribosomes are made of RNA and protein. The messenger RNA is read between the two ribosomes. And in the ribosome, we're getting a little bit into HL now. There are two binding sites in the larger ribosome, and the transfer RNAs bring the amino acids of the protein into these binding sites. Sort of. This is the syllabus. I think it's the way it's taught. There's the APE. E's for exit. So really, you have two sites. You have the A and the P. So you have the first transfer RNA comes together. So you have the A. Okay, so I'm not drawing. So here you have the A site. You have the P site.
Concentrate.
So what happens is the transfer RNA actually looks like a T. So basically, the transfer RNA looks like a T. So this is your transfer RNA. And at the end of the transfer RNA, this is your amino acid for your protein. And so each end of the transfer RNA has an amino acid, and they're going to bind together to make the protein. So the protein is somewhere over here. The protein is growing out of the ribosome. Now, I believe I'm on the verge. I'm on the verge between the two. HL needs to be able to explain all the steps. SL just needs to know like what transfer RNA is, messenger RNA is. I usually know these things are. So what is the messenger RNA? Where did the messenger RNA come from? It was complementary to the DNA molecule. And then the messenger RNA goes to the ribosome, where it's read and made into a protein. And the transfer RNAs bring the amino acids to the ribosome. Right there, SL stops. HL needs to know the finer details. And so the 3 locations, like E is for exit. So there's basically 2 locations. So one comes in, they bind, and then that one leaves, it shifts over, next one comes in, and so that's why some books say 3, some books say 2. I was always taught 2. But when I read the IB Oxford guide, it has 3. That's the first time I saw a book that said 3 locations. So I think it depends. It'll be something we could look up if you want to know. But basically, it's called indexing. So you have one comes in, one goes out. Okay? All right. I think I'm almost done here. So transfer RNAs, definition of transfer RNAs is they bring the amino acid. So write that down somewhere. Transfer RNA bring amino acids to ribosome. Okay? Messenger RNA is read by the ribosome. Okay? The ribosome is going to make the protein. Some other just basic vocabulary words. Okay, so if I say replication, I think we all know what replication means, right? What does it mean to replicate? Make an exact copy, right? So replication is DNA to DNA. Transcription is what the DNA to RNA does. So this is DNA into RNA. And the last one is translation. Translation is RNA into protein. All together, this is called the central dogma. The central dogma of biology. Dogma is like pillar, like it's a foundation, like the Bible is the foundation of Christianity. The central dogma is the foundation of biology. DNA is read into RNA. RNA is translated into protein. Transcriptase would be an enzyme that reads DNA. Something like that. Reverse transcriptase would be RNA to DNA.
Like AIDS or something?
We use this there are some, so first of all, that comes from viruses. Some viruses are DNA based, some viruses are RNA based. So an RNA virus, a retrovirus, has reverse transcriptase because an RNA virus will infect the cell with RNA, it will be converted back into DNA, and then DNA, RNA, protein to make virus proteins and reproduce. I lost you, Bonnie, huh?
Yeah.
What are you saying, Mr. Wei?
What am I saying? Well, that's why I'm recording this. So, let's review. What's DNA made of?
That.
What is DNA made of?
3 things.
Well, okay. You guys are saying the part. Okay, if I said what's DNA made of, I would just simply say nucleotides. Right? DNA is made of nucleotides. RNA is made. Okay, you could say like, what is DNA made of? DNA is made of DNA nucleotides. RNA is made of RNA nucleotides, right? What are the 3 parts to a nucleotide? Phosphate, nitrogen base, 5 carbon sugar. Okay, so we confident with this? Okay, so now we go on to DNA versus RNA. So DNA versus RNA, the big difference is what? The first big difference is what? I heard 2 things. I was going to say the sugar, but
The oxygen.
so deoxyribose is a sugar for DNA. Ribose is a sugar for RNA. DNA is double-stranded and RNA is single-stranded. The bases, the nucleotides in DNA are ATCG. And the bases in RNA are AUCG. Minor things, DNA is generally inside a nucleus or in a chromosome, right? So prokaryotes have a circular chromosome, right? So DNA is in a chromosome, right? It's in the nucleoid or the nucleus. Prokaryote versus eukaryote. Yes, sir?
What's the difference between a cytoplasm and a plasma? Or can they be used interchangeably?
Usually, okay, so when you say you're saying plasma, right? Or cytoplasm, or
What's the difference?
well, when I hear someone say plasma, my first thought is actually blood plasma, not like there is no like plasma of the cell. I've never heard anybody.
What's another word for like the cytoplasm? Because I remember there being another word.
I can't think of another word for cytoplasm, but cytoplasm is the empty space. Now, there is a specific name for cytoplasm of the mitochondria. So the cytoplasm of the mitochondria is called the matrix, and the cytoplasm of the chloroplast is called the
Ganglion.
no. You're thinking the membranes. You're thinking the granules.
What are you asking?
Okay, so in a chloroplast, so the mitochondria, the empty space is called the matrix. In the chloroplast, the empty space is called the S.
Sigma.
Stinky.
What are the holes in the bottom of a leaf?
Oh, stomata.
Yeah, but it's not stomata, it's the
Stoma.
stroma.
It's like domain.
Okay? So cytoplasm is empty space. Okay.
Oh, sorry, plasmid.
Yeah. Okay, so now we get into how DNA so now we're talking about DNA, RNA, protein. So when DNA replicates, that's DNA to DNA. Then we get to transcription, which is DNA, RNA. And then we get to translation, which is RNA to protein. Okay? So can everybody please give me the complementary sequence to this DNA molecule? Take a moment, write it down. What's a complementary sequence to this DNA molecule?
Write it down.
Oh, I didn't do that, did I? Chargaff's rule.
What?
Okay.
Equal.
Man, it's gonna be okay.
Equal.
A pairs to T, C pairs to G.
Yeah.
Okay? RNA for RNA, right? So this is DNA, right? For RNA, A goes to U, T goes to A, and C and G are interchangeable. So this is DNA to RNA, and this is DNA to DNA. Any questions?
No.
Okay, so there's your sequence. What is the complementary sequence to this piece of molecule?
K,
T, A.
K,
Okay, so T,
A,
A,
J, K,
A,
A,
G,
K,
T,
C,
C,
that T.
okay.
Wait, sir, what's a plasma membrane?
That's the same as cell membrane.
What's a plasmid?
A plasmid is a small piece of DNA. A plasmid is a small circular piece of DNA. This is a politician moment. Shh.
Politician, yes.
Guys, this is a politician moment. I would never say that that plasmids do not exist in eukaryotes. I think that'd be a falsehood to say that. But generally, you find plasmids in prokaryotes and not eukaryotes. But to say that no eukaryotes have plasmids, I think would be false. Because there's always an exception somewhere. So generally, yes, plasmids exist in prokaryotes, and because eukaryotes don't need plasmids. Eukaryotes have chromosomes. Okay?
Yeah.
But can a eukaryote be a single-celled organism?
Isn't it giant?
But can it be? Yes or no?
Yes.
So just no question, right? So it would stand to reason that some single-celled eukaryotes might not have chromosomes. They might just have like plasmids and one piece of chromosome. There might be something in between, right? If you're talking about a plant cell or like all plant cells, animal cells, all those have chromosomes and have large genomes,
Yeah.
right?
Yeah.
But single-celled organisms that are eukaryotic might only have one or two chromosomes, might have one chromosome, they might have plasmids. I don't know. I don't know every single. I don't have them all. I don't have the tree of life memorized. I'm not an encyclopedia. Okay, so can somebody please transcribe this sequence for me?
Into DNA or
Stop right there. I said I'm transcribing. I said if I said replicate this sequence, what is the complementary, right? I said, can someone please transcribe it for me? Which means DNA to RNA. So if I said transcribe this sequence, what is the complementary side? A, C, C, U. You guys sounds like zombies.
I was correct.
They're zombies.
Okay, so
Jeez, John.
now this is my messenger RNA. Okay? The messenger, so when you talk about RNA, there's three kinds of RNA. There's transfer, messenger, rRNA. So the messenger RNA goes to the ribosome, and it's read by the ribosome, and it's translated into a protein. And that's probably what confused you with the whole, like, there's transfer RNAs. So in a ribosome, here's my picture. In a ribosome, we have transfer RNAs. Transfer RNAs have an amino acid attached to them. Okay? And this is where I'm actually going to pause the video and just show you photos from like the website. It'd be easier if I show you photos.
Just imagine this entire